Ever wonder how a newborn mouse, or you once upon a time, builds a working brain without a blueprint taped to the crib? Right now, as you're listening, billions of your own neurons are still following an internal GPS they never had to memorize. No teacher, no turn-by-turn instructions, just a genetic code whispering, "You, over there, connect to that spot." Today we're cracking open that secret navigation system. Here's the roadmap for the next 10 minutes. First, we'll zoom in on what scientists actually measured. Then, we'll see how those measurements reveal an address label for each neuron. Finally, we'll explore why that matters for learning, for disorders, and for future therapies. Sound good? Let's dive in. Section 1. Finding the fingerprints. Picture a lab scene. Tiny samples from a mouse brain, thousands of cells sorted like beads of light. Researchers switched on a gene sequencing machine to read which genes were active in every single neuron. At the same time, high-resolution tracers followed the long axons those cells sent across the brain. When they poured that mountain of data into machine learning models, a pattern snapped into view. A cell's gene profile predicted, with surprising accuracy, the exact neighborhoods its axon would visit: motor zones, sensory hubs, memory centers. It's like glancing at someone's playlist and guessing not just their favorite genre, but the club they'll hit on Friday night. Section 2. Which genes matter? Now, you might ask, is there one master gene steering the ship? Not quite. When the team sifted the data, a short list of guidance genes popped out, molecules on the cell's surface that sense attraction, repulsion, or a place to latch on. Think of them as colored stickers on a suitcase. One sticker says, "Avoid the midline." Another says, "Stop at layer 5." Alone, each sticker is helpful. Together, they form the baggage tag that gets the neuron to its gate. And here's the twist. Change just one sticker, dial its expression up or down, and entire routes shift. Earlier animal studies showed that knocking out a single guidance gene can make whole bundles of axons veer off course. The new work explains the subtler side. Tiny differences across many genes fine-tune even neighboring neurons so they don't all end up at the same party. Section 3. Timing is everything. Let's pause for a breath. Guidance-related genes don't stay stuck on high forever. During development, they rise and fall in waves, each wave lining up with a fork in the road. Should the axon cross to the other hemisphere? Dive to a deeper layer? Branch out for multiple targets? So the code is less a static address and more a sequence of green and red lights that open one lane while closing another. That time-locked sequence matters when things go wrong. Instead of too many or too few connections, many neurodevelopmental conditions show misdirected ones. Healthy axons, wrong destination. Large human genetic studies back this up, repeatedly implicating guidance pathways in autism, intellectual disability, and more. Even a modest detour can cascade, changing which signals meet and how they sync. Section 4. What does this buy the brain? Okay, zoom out from single neurons to whole skills. When a mouse learns a new movement, the highway from planning zones to the spinal cord doesn't appear overnight. It's pre-surveyed by this genetic GPS. Learning just adds new lanes, closes others, or repaves the asphalt. That logic may explain why one brain feels ultra-flexible while another seems brittle. Tiny shifts in those molecular addresses might give person A extra backup routes and leave person B with only one narrow bridge. Disorders tied to guidance genes might not delete roads at all. They just send traffic through odd intersections, forcing signals to crawl along the scenic route. Section 5. Future tools and therapies. Here's where it gets exciting. Imagine real-time traffic cameras for the brain, tools that watch axons as they settle. Subtle route changes could become early warning flags for learning issues long before behavior shows it. Therapies could then nudge axons during the short windows when detours are easiest. Down the line, synthetic circuits might swap in alternate links when stroke or trauma closes a major corridor. Section 6. Where we're headed. We're only at Chapter 1 of this map-making saga. Next steps include matching wiring codes to memories forming, habits breaking, or skills returning after injury, like tracking footprints in fresh snow. As tools sharpen, scientists may not just observe these routes but gently draft alternate paths for the brain to try out. Recap. Let's land the plane. So, what did we cover today? Let that sink in for a second. 1. Your neurons carry a built-in GPS, combinations of guidance genes that act like address labels. 2. Those labels are dynamic. They flip on and off in sequence, steering axons through decision points. 3. Small tweaks in the code can reroute traffic, shaping learning capacity and sometimes contributing to conditions like autism. 4. Because the map exists before experience, future therapies could read, predict, or gently redirect these routes, opening new doors for recovery and circuit design. Here's the empowering part. The next time you practice a skill or recover from a setback, remember, you're working with a city that's already surveyed but never finished. Streets can widen, shortcuts can appear, and with the right cues, or maybe, one day, the right gene tweaks, traffic can find smoother paths. Call to action. Between now and our next episode, try this. Pick one topic: autism, spinal cord repair, or neurodegenerative disease. Search for a recent paper that mentions axon guidance. Skim the abstract. Ask yourself, which molecular sticker on the neuronal suitcase is the study focusing on, and what route might change because of it? Then, share that mental map with a friend. Explaining it out loud will cement the concept in your own neural network. All right, travelers, that's our tour for today. Keep your curiosity fired up, and remember, every thought you have rides along a route that genes first surveyed. Until next time, keep exploring those hidden maps.